Hello dear learners, today we are going to discuss the MCQs of Microbiology Part IV from the bacteria, which mainly focuses on staining of bacteria. In earlier videos for bacteria Part I, Part II, Part III, we have seen few of the mixed sort of MCQs on topic bacteria, and bacteria 4 majorly covers staining. So to start with the first question from the staining, the fimbriae are demonstrated by, whether by culture, whether by gram stain, whether by biochemical reaction or hemagglutination test. So before proceeding, we will see what do you mean by agglutination, what do you mean by hemagglutination. So agglutination is nothing but a simple adhesion process in simple words, whereas hemagglutination, as you can see here, RBCs. Hem means RBCs and agglutination. So it is adherence of RBCs which is macroscopic in nature, means we can visibly see this hemagglutination step. Antigen antibody complex formation takes place. Now, as you all know, fimbriae, why it is used for fimbriae demonstration? Fimbriae are short and half thick than flagella like structures which contains a protein called as pillin. So this pillin protein, as you all know, antibodies which are also known as immunoglobulins are nothing but a large Y-shaped protein produced. So this antigen antibody complex will give you the results. So this way hemagglutination is nothing but accumulation or adhesion of RBCs which will give you the visible result. Correct answer for this is, it is hemagglutination test. So fimbriae can be demonstrated by hemagglutination test. Next question is, bacterial spores are, whether they are weakly acid fast, whether they are strongly acid fast, whether they are alcohol fast, or they are non-acid fast. So the correct answer is bacterial spores are weakly acid fast in nature. Next question is, endospores can be stained with, whether it is with safranin, whether it is with crystal violet, whether it is with methylene blue or malachite green. The correct answer is, it is malachite green, because if you will see the spore staining, then it is a primary stain which is nothing but malachite green. On application of heat, it will act as a mordant, that means it will fix the stain and spores will get the stain of malachite green. Now application of water will act as a decolorizer, and this bacteria, majorly the gram negative bacteria which are there, or it is specifically spore structure is found in gram negative only. So these bacteria will lose this primary stain, and that is on application of decolorizer, and they will catch the, or they will capture the counter stain, that is safranin, which is secondary stain. So the remaining part of bacteria other than the spore will get stained by secondary stain or counter stain, that is safranin, and spores will remain stained with the primary stain. Since, as you all know, we have seen the spores type, so the location is like terminal, then subterminal. So as you can see here, the terminal green colored structures present in the bacteria are nothing but spores which get stained with the primary stain, that is malachite green. The next question is, the order of stain in gram staining procedure is, whether it is crystal violet, iodine solution, alcohol or safranin, whether it is iodine solution, crystal violet, safranin and alcohol, whether it is alcohol, crystal violet, iodine solution and safranin, or all of these. The correct answer is, the sequence is crystal violet. First is primary stain, then mordant which fix the stain, then alcohol as a decolorizer, and then secondary stain. So the sequence which you can see is crystal violet, iodine, alcohol, safranin. So the correct answer is crystal violet, iodine, alcohol, safranin. Next question is, percentage of alcohol used in gram staining is whether it is 75%, whether it is 90 to 95%, whether it is 60% or 25%. The correct answer is, according to the steps, if you will see, the alcohol which is added is 95%, that is 90 to 95%. Next question is the percentage. So the correct answer is, it is 90 to 95% of the alcohol is used in the gram staining. Then next question is, gram positive bacteria appears as, whether they appear as pink, violet, both A and B, or none of these. The correct answer is, it is violet. The question is, gram positive bacteria appears violet. Why they appears violet? Because if you will see the diagram, according to the staining technique, this gram positive bacteria, even after the action of the decolorizer, they will not lose the stain and they will remain stained with the primary stains color, that is purple or violet. So these are here you can see purple colored gram positive bacteria, and this pink color which have been stained with the secondary stain, that is safranin, are gram negatives. So it is violet color or purple color. Next question is, the action of alcohol during gram staining is whether it allows the color, it adds the color, decolorizes the cell, or none of these. The correct answer is, it is decolorizes the cell. The alcohol which is used as the decolorizer in gram staining, it will decolorize the cells just for the identification purpose. Next question is, lipid content is more in, whether it is gram negative bacteria, gram positive bacteria, both, and none of these. So the lipid content, as you all know from the structure of the cell wall, it is more in gram negative bacteria. So lipid content is more in gram negative bacteria. Next question is, the cell wall is thick in gram positive than gram negative, thick in gram negative than gram positive, equal in both. In gram negative, cell wall is absent. So the correct answer is, it is thick in gram positive and than gram negative. So cell wall is thick comparatively in case of gram positive than the gram negative. Next question is, the question says, the lipid content present in the gram positive bacterial cell wall is, whether it is 10%, whether it is 1 to 5%, whether it is 20%, or none of these. The correct answer is, it is 1 to 5%. The lipid content present in gram positive bacterial cell wall is 1 to 5%. Next question is, if only one stain is used for staining a specimen, then it is a simple staining, negative staining, differential staining, or none of these. The correct answer is, it is simple staining. When only one stain is used, it is known as simple staining or monochrome staining. Then next question is, other than the sample specimen, the remaining portion is stained, then it is called, means background will be stained and the sample, the actual point or actual microorganism which is there, that will remain colorless. So such kind of staining, as you can see here, the background is colored and the organism of interest is colorless. So the question, the answer for the question is, it is negative staining. That means other than the specimen, the remaining portion is stained, then it is called as negative stain. Next question is, during staining for electron microscopy, the method which improves the contrast of specimen is, whether it is positive staining, negative staining, shadow staining, or none of these. The correct answer is negative staining. During the staining, the electron microscope, for electron microscopy, the method which improves the contrast of specimen is negative staining method. With this, we finish with few of the MCQs from the part bacteria, that is fourth part. which focuses on staining. Thank you.